I'm Michelle Boxall and welcome to Behind the Eight Ball. When the odds are stacked against you, how do you get out in front? Well, in IBTV's new series, we will hear about the failures along the road to success for many of the advanced therapy sector's esteemed CEOs and founders. We hope we can all learn from these stories and that they will provide inspiration for new and future successes for the industry. So with pleasure, I'd like to introduce Dr. Luigi Naldini. Luigi is our third Behind the Eight Ball interviewee after Claudia Zilberberg and Sven Keeley, and a really super special CGT industry heavyweight. We were really excited about being able to support the announcement on your Lifetime Achievement Award on behalf of Facilitate. Luigi, you're very welcome.
Thank you very much for hosting me, and thank you for also supporting the award story. It was really a great honor for me to take that.
Yeah, it was a great accolade. So we'll jump straight in. Luigi, today you're a professor of cell and tissue biology and of gene and cell therapy at the San Raffaele University School of Medicine and scientific director of the San Raffaele-Telethon Institute for Gene Therapy in Milan.
In Milan, Italy.
Some of your accolades, which there are too many to list, but obviously the main ones, you pioneered the development and the applications of lentiviral vectors for gene therapy, which have become one of the most widely used tools in biomedical research and have allowed a generation of several gene therapies, some of which have been approved for the market. You're obviously also co-founder of 3 biotech businesses. Looking back, what do you think set you along the path and onto these many accomplishments? Are there any early stories that give us some insights into the person you've become?
Well, I studied more classical high school, which in Italy is more like in humanities. And then I became very fond of science, and I started to really deep into physics and then biology. And I think at that time was really when biology was becoming a new science. So we were learning about DNA and RNA and the translation. And I became so fascinated by all this new world that I really decided to focus on that. And so I essentially decided to take that career. Mostly, I think, driven by curiosity. And I thought I would become a scientist and really delve into curiosity-driven research. But then the path to research, especially at that time, I was mostly advised to go to medical school.
Yeah.
So that's and then to a certain extent, I think that mix, background. So on the one hand, the deep interest for basic biological model and system. At the same time, however, learning about medicine, I think it was really, to me, the key, setting me up on this path in which science for translation into medicine. So keeping, on the one hand, always an eye on the basic biological process, but then try to understand and manipulate them for development of treatment. So I think that really came during my education.
That's really interesting. So I've noted a lot, when I speak to industry leaders and really successful people, they often talk about having a curious nature. So that's not surprising. Before we talk about your 8 Ball story, and you look back, could you tell us a little bit about how you felt about failure? Were you scared of it? Did you not think about it? Were you, did you have just an eye for success?
No, I think failure is something you face, and I think you prepare for it in terms of building your knowledge and also your self-confidence. And of course, this is a process, right? I think I set always pretty high bar for me during my early times, both in study, in the term of achievement at university. And I, of course, had also challenges. I had some failures that teach you not to stop there. but just to start again and learning from that. I mean, maybe that time you didn't go enough, you didn't spend enough time to learn about that, or maybe your attention was not sufficient for that particular challenge. And I think this is something you learn. If you have a goal, which you clearly have in mind, I think you have to get there, and then you see challenges and failure as a natural
part of the progression.
part of the progression.
Yeah,
Yes.
part of the process. So, really excited to hear your 8-ball story, a time when, Luigi, you failed or nearly failed.
So I think my key story is really this effort to develop the gene therapy vector, which I started to work on in an academic environment at the Salk Institute. It's almost 30 years ago by now. And again, after really try to translate my effort to understand biology, but also to develop new medicine, gene therapy really looked like the ideal and enterprise. And this was early days, and there was a key need to generate new vectors which were more powerful for achieving the gene transfer goal. And HIV had emerged, unfortunately, as this new virus. It was a pandemic at that time, unfortunately very tragic for the number of people died. But this new virus, which has a lot of features which could have made a very good vector if we could able to tackle it, and of course get rid of some potential pathogenicity. So I worked on with that couple of years at the Salk Institute. We laid the foundation of this new vector. We made a very beautiful paper in Science, which was a key achievement. And then I was essentially at that point, I was asked to move into a biotech sector because there was interest from biotech in the Bay Area to develop this technology. My original plan was to come back to Italy, actually, in academia, but then I changed completely my plan. I thought my opportunity was to follow up with that, to really make this a real tool. I mean, moving from a proof of principle at the bench into a real development. And so I spent another 3 years or so in the biotech industry to really move this forward, not without challenges. But these are not the ones I'm talking about. There was challenge because, of course, working with a vector derived from HIV was more than scary and unlikely. So several times I was asked, well, you did a good job, but are you really serious about that? Do you really want to make a vector? Do you think we'll ever be able to put that in humans, and so on? So I think I really trained in terms of confidence, and I learned really to push through because I had a strong, of course, belief in what we were doing. And then I think the climax was when at a certain point we were really almost ready to start thinking of the first clinical trial with the company. And unfortunately, what happened was the first announcement of a severe adverse event in a gene therapy trial in Europe, which had not been using HIV vector because that was not available yet, but a similar vector derived from a retrovirus. And although initially it had worked, these people developed leukemias. Right. Which were ascribed to the vector insertion. And this was a major blow to the whole field because, of course, this was a risk that was not anticipated in reality. There could have been, to a certain extent, concern about that, but the whole attitude was that this was not something it could have happened in the setting of a vector. And so from that day to the following, essentially, the company decided to halt the process. It was too risky. I was called by the CEO. I said, this is too much for us to continue to develop something because the field lost trust. So not only we had the issue of a very dangerous virus we started from, but now we had to face loss of confidence from the whole community. And so essentially, at that point, that was really the biggest risk was to have everything would have stopped there. And after all this, now it was 5 years of hard work, all this effort to move forward, and it could just go away because there was this general loss of confidence, as I said, from the community. And so, well, of course, there was not much I could do there.
Like it's out of your control.
Exactly. And also in the company, there's issues there. Of course, this is more relatively small company. They couldn't afford further to move forward. And so I decided to go back to academia. I went back to Italy at that point. There was a position of a professor. I took as much as possible with me of that work. I remain, of course, linked to the company for a couple of years, trying to salvage what I could.
Yes.
And then I connected to a charity, Telethon you mentioned. It's a charity for really supporting research on genetic disease, and try to convince them that there was a value in what we were doing. And so essentially, progressively, we rebuilt the potential to move forward. So this was not anymore the setting of a biotech, but it was an academic environment with a strong support by a charity. We decided to put a lot of money into continuing that effort. But then I think the key challenge then was, of course, to overcome that loss of confidence with the scientific community, which at this point was sort of rejecting gene therapy, say, there was a lot of hype at the beginning, and then, of course, now we don't trust anymore. And so I think, again, what has been crucial there is that, and it took a couple of years, of course, to do that, to go back to the basic. And we started in the lab, and we said we should question why there has been this failure to predict the risk in the clinic with this early generation, early vector. And can we show that the vector that we have generated now, paradoxically, because it's coming from HIV, is safer? And of course, we said if we don't show that, if we don't prove that to a certain level of evidence, there's no way we can go forward. And so essentially, we really start to think about why this was not, this risk of leukemia was not really predicted, or at least emerged in preclinical testing. And there was some reason that, in retrospect, were very obvious. A mouse has a short life as compared to humans. Those leukemia emerge a few years after treatment. Of course, you don't test as many cells in the mice as you test in humans. And so overall, I think there was not sufficient preclinical testing. And the question was, how can we do it now? Because if we do the same, we know that we have no way we can show that our vector is better or not. And so I think one of the key ideas was, let's take mice which are tumor prone. So mice which are very sensitive to the risk of developing leukemia. And let's challenge them with this gene therapy vector. And let's challenge them with the old vector which gave problem, and the new vector that paradoxically derived from HIV. We work so much on it to make sure that it is safe, that maybe it will be safe. Yes. And then we started to see that that's actually those mice which were, they were tumor prone, so they would develop tumor. But if we treated them with the old vector, they developed tumor before than the other groups. And we saw that the tumor they were developing had the vector inside, so there was a causative role. And when we put the new vector derived from HIV, this was not different from the control group. And so I think we started to build the real evidence, scientific evidence, that there was an advantage of the new vector. And this was one of the key, I think, proof, and we started to of course, report to the scientific community, together with the proof of efficacy of the diseases that we were trying to working on. And it was not easy, because I remember people at the audience, in fact, like here, of course, many years ago, say, how can you say that your vector is safe? All the mice are developing tumor anyway. And they said, well, they do, because they are tumor prone, but we are looking at a different time. And I think that was, scientific evidence of that really allow us to overcome this big, of course, time of skepticism, and provided an additional rationale to move forward into the clinic. I mean, in principle, we could have also avoid doing that. We could just have pushed through the strategy, because we had good preclinical evidence of safety in a mouse model of the disease that eventually we started to treat. But I think having so thorough, try really to address the issue from the basic root, and I think allowed to overcome. And what have been, if you want, a failure, which was ours, but also a failure of our field in general, I think it allowed us to really overcome it.
Yeah, and it's very much speaks to the previous question when you talked about your approach to failure and not really being an approach to failure. It's more, this is how you need the failures to progress, and that's basically your story as well, isn't it? Where you've gone back and had to then find more proof points and more data and do more research from different perspectives. But it's really interesting to me because you spoke about the biotech situation and having to move on from that. But it seemed as though you didn't, you just thought, oh, I've got to move on, improve. It didn't seem like there was any lack of confidence. There was more the fact that, okay, I need to prove and find a way forward. You didn't ever think, actually, this gene therapy business.
No, I never thought that, because of course this also was driven by evidence. I think we had evidence in our studies on preclinical modeling, and we were curing mice of the disease. We were seeing the mice were doing well. They were not getting sick, not only for the disease, but they were not only also getting sick from the treatment. And so we have this tool, which was, the original goal of developing this vector was to have an efficient vector. And this was efficient, and it was efficient, not only in our lab. There was a whole community were using that vector in experimental modeling. And so we were saying, well, this thing works.
Yeah.
And why not giving a chance to get to the clinic again? And so again, I think it was, of course, you had to be stubborn to a certain extent. But again, not because this was your idea, but because I think it was every day there was a hard check with the reality. I always thought that if we had seen a failure in the preclinical model, if we had seen a risk emerging,
Yes.
We would have to stop.
Yeah.
And so again, we didn't want to be blind and not just say, no, you don't want to see. I think the approach was say, let's go and see as deep as possible. If something doesn't emerge, we have done all the possibilities that we could think of, and that, however, build our confidence for moving forward. And it's important because if you are the first one to do something, you have a huge responsibility, more than in other situations, because you have for the whole, of course, platform, for the whole technology. And so you have, maybe it take longer. Of course, this took longer. Of course, we didn't cut corner, and we didn't, we took, overall it probably was 10 years of work.
Yeah. Well, I think that's such an inspirational story for the current industry. Usually I go on and my next question is, can you speak about your successes now? But we've obviously, from that story, you've kind of talked us through how you then moved on to success. But did you want to add a little bit more about the successes?
Well, the success eventually came because this trial that we eventually started has been extremely successful, even beyond our expectation, I must say. And this is a drug that today is available on the market. In fact, it was approved in the US also not a few months ago. It was approved 2 years ago in Italy. So we have the amazing finding that we can offer a treatment to children who before had no hope. And this has been reproduced and repeated so and so. So, and again, I must say this also came because eventually pharma came back. So eventually when we had the first proof of principle in the clinic, we went back to speak with, at that time, big pharma and caught their attention again. And again, it took a while again, and I think it was the confidence now built again on the science, but then it was also the first proof in the clinic of initial clinical data. And then at that point, the pharma got back, and of course, gene therapy has become a reality now. But successes come and challenge comes again, because now we have success, we have effective treatment. The biggest issue that we are facing is that this treatment may not be available to most of the patients in need because they are too costly. And so this is not, we don't have a solution yet because these are costly treatment. And unfortunately, the market has set a very high price. And our biggest concern now is that this treatment may not be available to most of the patients in need. And so that's another big challenge. Even beyond us as a scientist, it's really a societal challenge.
Yeah. I mean, an industry challenge too. So moving a little bit on to, how you lead, because obviously, you can be the best scientist, come up with the best ideas, but to really, truly be successful, you need to galvanize a team. What type of leader are you?
Well, I think it's, I think a key point, of course, is share the sense of a mission. So have a clear goal in mind that you share with the people. Feel everybody part of and responsible for. I think as much as doable as possible, I always try to involve even the most junior or the latest acquaintance to the team, to the overall goal, and to see the big picture, not using people just because they are instruments. Of course, all of this requires large team and different expertise. I think my period in the industry has helped me a lot to understand the relevance of teamwork, and also to how important it is to credit and value different expertise that are needed when you move into translational and eventually clinical trials. So this concept of leading a large team and making sure that everybody feels to be a very important part of it, from the most junior to the most senior, they all have to be part of this. And then I think a certain level of human relationship as well. We are not just mechanics working on something, but human beings. So there is a lot of that. So working relationship must be relevant and also responsible to take into account that these are all human beings that have. And so I always thought that I think that the environment around what we did was pleasant. Of course, people had to feel some pressure, but that doesn't mean they have to be stressed necessarily. They usually, you see them. I think I manage always to have a very strongly collaborative team, and this is of course one of the best. Reward that you have, to people that the people working with you that like you, and I think that's very crucial.
That's good to hear. And then, in terms of, obviously, we've had so many insights while speaking. If you were to give the industry one piece of advice that's always kept you in good stead, what would that piece of advice be?
Well, I think, again, probably from my experience is really to continue to pay attention to basic science and new ideas, emerging concepts, because they, of course, bringing the solution as well. And often, of course, those solutions and those emerging concepts don't come from the field you're working on. And so I think to be open and listening to what happens more widely in the field is crucial. It's crucial, of course, for a scientist in academia, for a young trainee, but it's also crucial, I think, for industry. Not to be too looking closely at that specific sector, because the new solution come from the unexpected, of course. Everybody knows the expected things. So I think to keep that mind open, of course, to different level, advisors, attention, for us coming to meeting and going to listen, not the usual people, not the usual story, but also the new one. So this is probably, again, this is coming back to my original curiosity which I still have, but I think this is a key driver for innovation, right? That's where you can catch new ideas. And I think this is true when we talk about innovation in the industry, novel medicine, these are not obvious things that we are doing, and you need to be creative, to think out of the box, also for development, also for regulators and so on. So this is probably my clear probably message from my experience.
And final question, looking forward, do you have any more goals or There could be personal goals that you still, things that you still want to do.
I'm still pretty active because I think we still have a lot to do in terms of this strategy that we have been developing. For instance, moving from the rare genetic diseases where most of this has been developed into more, common and acquired cancer. We have this tool to treat those, and so trying to make the proof of principle that this treatment are valuable also for more broad diseases, because we have an immense power in terms of genetic engineering. So this is something I still want to continue to move, to put some additional, let's say, step forward. And the other thing, of course, is more like, this institute we have been building over the years, we've been growing more and more. I'd like to, again, to let it in the best, for the future, right? I mean, to really build the foundation for the next step, which will not be my work. It will be the work of the younger people, the new recruitment who are there. But I would like to make sure that they start in the best manner. So try to use what has been success that I have been lucky to achieve it, use them to launch the next generation.
Yeah. Well, you just used the word lucky, and I'm going to use it as well. I think I've been really lucky to have had this time speaking to you. It's been a really super special interview. I think lots of the industry will learn a lot. And yeah, thank you for your time.
Thank you. Thank you very much.